I'm very happy. I'm very happy to have the honor to introduce Elisabeth here today. I first met Elisabeth in 2009 when I actually was doing my bachelor in the lab of Dietrich Ober, and when she was finishing up her PhD. And I remember many really good discussions with her about different topics from politics to science. And scientifically, Elisabeth had influenced me a lot, and especially her interest in enzyme and structure-function relationship has been always very inspiring for me. She is using the Pyrrolizidine alkaloid pathway as a model systems to study these protein enzyme evolution and structure-function relationships. And she has published her work in different high-impact journals. I just have a couple up here. And yeah, I'm very excited to hear her most recent research. And thank you, Lizzie, for being here today. And the stage is yours.
Yeah, cool. Thank you, Lars, for the introduction. So I'm going to share my screen with you. So, and it should work. You should see my screen. And perfect. So, yeah, as Lars mentioned, I'm studying the evolution of pyrrolizidine alkaloids, and I focus on the plant family of the Convolvulaceae or morning glories. And yeah, the story that I'm going to tell you today shares many characteristics with the evolution of caffeine biosynthesis that was presented, I think it's 2 weeks ago now, in our first talk. So there are many, they have many things in common. But before I go into detail, I will start with some introductory slides. First of all, very short, what are pyrrolizidine alkaloids? Well, they are secondary or specialized metabolites. More than 400 structures are described up to now, and they are part of the plant defense against herbivores. All the PAs share a common core structure, which is highlighted here in red, the Nesine base. And the huge diversity of pyrrolizidine alkaloids, or short PAs, comes from an esterification of the Nesine base with a variety of acids, which then are called the Nesine acids. Okay, so that's how pyrrolizidine alkaloids look like. The biosynthesis of PAs is initiated by the Homospermidine synthase, and this enzyme catalyzes the transfer of spermidine, the transfer of an aminobutyl moiety from spermidine to putrescine, and thereby it produces homospermidine, and then further enzymes convert homospermidine directly to the Nesine base, so to the common core structure of PAs. So this is a key step in pyrrolizidine alkaloid biosynthesis, and it connects the primary metabolism of plants, so the polyamine pool are part of the primary metabolism, with the secondary metabolism. Okay, and unfortunately, this enzyme, the homospermidine synthase, is up to now the only known enzyme of PA biosynthesis. What we also know about this enzyme is that it evolved by a gene duplication event from the deoxyhyposine synthase. And this enzyme is a very conserved and a ubiquitous and very conserved and ubiquitous enzyme of primary metabolism of plants, but also so general eukaryotes, also humans have a DHS, and it is involved in the activation of the eukaryotic initiation factor, and this initiation factor is important for the regulation of cell growth. So in humans, it was shown that the AF5A and the activation of the AF5A is important also in inflammatory processes or in cancer development. Okay, so this, the deoxyhyposine synthase in blue, is the ancestral enzyme. And so the ancestor and then the derived enzyme, the HSS, they share the reaction mechanism. Both transfer an aminobutyl moiety from spermidine to a preferred substrate, and they just differ in this substrate. So this functional change can also be interpreted as a, well, in the end, it's a shift in substrate preference. And what I also need to mention here, or I want to mention, is that the ancestral enzyme, the deoxyhyposine synthase, can accept also other substrates. It can even accept putrescine as a substrate, and also can produce minor amounts of homospermidine. And in that way, we can say that the DHS, the deoxyhyposine synthase, is a promiscuous enzyme. And this is also something that we heard in caffeine biosynthesis. So the enzymes, the ancestral enzymes are very often promiscuous. Okay, and then what is also interesting, this gene duplication and the recruitment to PA biosynthesis occurred in many different plant families. So we have a broad convergent or parallel evolution of pyrrolizidine alkaloids. Also something that we heard for caffeine biosynthesis. And yeah, this is a gene tree from sequences that encode homospermidine synthesis and deoxyhyposine synthesis from the different plant families. All the plant families they are, which are highlighted with the color code, these are all families that produce PAs. And in all of these families, there was an independent duplication event and then the recruitment to PA biosynthesis. And in the Asteraceae, we even have 2 independent origins of PA biosynthesis. Okay, so far to the introduction. I'm now going to focus on the evolution of pyrrolizidine alkaloids and homospermidine synthase in the morning glories. The morning glories are interesting because PAs in this family occur only in a very few species. And on the left side, you can see an illustration of the phylogenetic relationships of the major tribes of the Convolvulaceae. And PAs only occur or have been described to occur in the Convolvulaceae. And this is a zoom in now to the Convolvulaceae tribe. And then the PA producing species are highlighted with the yellow color. And so you can see we have 2 lineages that produce PAs. It's the Quamoclete group in the Ipomea lineage. And then we have the genus Distimarca, and also in this genus, a few species, in fact, only 2 species are described to produce PAs. And also interestingly, the PAs that are produced by these 2 different lineages also differ. So, and not only in these acids that I mentioned that are esterified to the core structure, the nicene base, but also the nicene base itself differs. So we have in the Ipomea lineage, we find saturated nicene bases, and then in the Distimarca lineage, we find these unsaturated PAs. And of course, if one sees this pattern, one hypothesis would have been that we also have an independent evolution of PA biosynthesis and an independent recruitment of the homospermidine synthase. And so this was basically the start of my PhD thesis. And the first step was to screen the diverse species, the Convolvulaceae species from the different lineages for HSS and DHS encoding sequences. And so we continued this screening for such sequences, and now I have a little larger data set than compared to my PhD thesis. And I can show you the data set that we have at the moment. And so these are DHS and HSS encoding sequences from the Convolvulaceae. I labeled the DHS clade here and the HSS clade, and I also indicated the duplication event. And yeah, interestingly, we found not only in the PA-producing species HSS gene copies, but also in many Convolvulaceae species that do not produce PAs, we found a putative HSS gene copy. So this was a bit surprising. And what do you want to mention? Yeah, also interesting, the gene duplication. According to this phylogenetic tree, the gene duplication event occurred prior to the divergence of the Distimarca and the Ipomea alba. And Miller published Eserman, they published a fossil calibrated phylogeny of this Convolvulaceae, and the divergence time of these two lineages was estimated to be 50 to 70 million years. So we can also conclude that our gene duplication event at least has this age. So it is at least 50 to 70 million years old. And this is a rather old duplication event, or in other words, the retention time of the duplicates is rather long. Based on the genomic data available, the half-life of duplicated genes was estimated to be 2 to 30 million years. And so these copies are at the, yeah, they're really old. Okay, and this broad occurrence of a putative HSS encoding gene prompted us next to reanalyze these species for the presence of PAs. Maybe they were overlooked in the past, and also, of course, our analytical methods improved over the years, and so we reanalyzed them with gas chromatography coupled to mass spectrometry, so GCMS analysis. And also we analyzed the above ground plant material, but also root material. And in the past, most of the studies focused in the above ground material, as it's just easier to sample, of course. And but we cultivated the species in our greenhouses, and so we also could analyze the roots. And on the next slide, I show you a typical GCMS profile from the Distimarca quinquefolius roots and the Ipomea alba roots. And on top, you can see Distimarca quinquefolius and also the classic PAs that we could identify in this extract. So the nesine base, the unsaturated nesine base, and in the chromatogram, most of the peaks here, or all of the peaks here, are really PAs. So the root of Distimarca quinquefolius is super rich when it comes to PAs. In the above ground materials, we really found much less PAs. The roots seem to be the organ in which the morning glories accumulate their PAs. And yeah, below this chromatogram from Distimarca quinquefolius, you can see the Ipomea alba roots, the chromatogram of these species. And compared to the, also I need to mention, Ipomea alba is the only species in which we could detect, only new species in which we could detect PAs. In all the others that we screened, no PAs were found, only the ones that have been already known. So Distimarca was known before our screening. And yeah, Ipomea alba, the only new species, and you can see only this tiny peak, which is indicated by the red arrow. This is a PA, And this seems to be a super, not seems to be, it is a super simple PA. It's basically only the nesine base on the right side. So the iso-retro-necanol, that's the one that we identified. And yeah, maybe Ipamera alba has a very primitive pyrrolizidine alkaloid pathway. Okay, so what is next? Yeah, so I added to these three a color code. Yellow means we have PA producing species. Green is the one with the nesine base. And then I also need to mention here the orange color. Algeurea nervosa produces lowline alkaloids, and they are structurally very similar to pyrrolizidine alkaloids. But it is it is assumed that they are produced via a different biosynthesis and maybe also produced by a symbiotic fungi. Okay, so I will not further discuss this lowline alkaloids. Yeah. Still surprising was that many species that don't produce PAs have a putative HSS gene copy. And the next question that we wanted to address is the function. What is the function of these genes? And we started with biochemical characterization of the encoded enzymes. We did some in vitro assays. And here I added the specific activity of the encoded enzymes for homospermidine production. And on the first, yeah, they clearly differ in their activity, the different enzymes. And so the highest homospermidine producing activity, we found it for the HSS of this demarca quinquefolius, a PA producer, and a different individual with a slightly different HSS. Here we had a very, very low homospermidine synthase activity, but we also found PAs in this individual. Also, I want to highlight Ipamera alba HSS, the one with the primitive PA. It has a very good homospermidine producing activity. Then here the HSS from Camonea umbellata. This enzyme does not produce homospermidine at all, so it seems to be not active. It also doesn't catalyze the ancestral activity, so it also doesn't accept the EF5A as a substrate. And yeah, presently seems to be a non-active enzyme. And then also, of course, the oxyhyposine synthesis, the ancestral enzymes, they have also a weak homospermidine producing activity. And yeah, this variation in the activity gets even it's getting more interesting when we consider the sequence similarity. And so this is something different to the caffeine enzymes that we heard last time. HSS and DHS are super conserved. They are still highly similar. They share around 80% sequence identity. And it is based from the sequence itself, it's really not possible to predict the function. There was just one exception. We and also other authors found this very specific pattern in homospermidine synthase encoding sequences, and not only for the morning glories, but also the HSS from the Asteraceae, which produce PAs, and the Boraginaceae, and so on. And all these HSS shared two specific amino acid exchanges from DHS to HSS. And what's the next slide here? I highlighted them here in this little alignment. That's the exchange from isoleucine to valine at position 277. And then there was another exchange to not really aspartic acid at position 281. And these exchanges or these sites, we also did some positives, we calculated selective pressure, and these sites are under positive selection in the ipomöa, in the PA producing ipomöa lineages. And a third site was positively selected. This is also indicated here, the site 266. And yeah, this pattern of these independently evolved amino acid changes in the very different plant families is on one hand interesting, and it was also suggested to use this motif, the VD motif, as a marker to predict the function of the encoded sequence. And so for 90% of the HSS sequences, this marker might work. But in our case, so in the case in the Convolvulaceae family, the marker is not associated to any of the to the function. So neither to the activity with the Homospermidine producing activity, because for example, ipomöa alba has a very high activity, but it doesn't have the HSS motif. And on the other hand, in the morning glories, it also cannot be used as a marker for the presence of PAs in a species. As here, the second individual from this demarca, quinquefolius, also doesn't have the motif. It only has the exchange here, so it only has the V of the motif. But this species, we found very nice pyrrolizidine alkaloids in the root. So I think the motif is very interesting as something that repeatedly happened during evolution has to mean something. I also, yeah, here with the blue dots, I mapped the ancestral activity. So the enzymes that show an activity with the AF5A. And of course, all the DHS enzymes have this ancestral activity, so they have a blue dot. And then in the HSS clade, only the ipomöa alba also has the blue dot. So the ipomöa alba is also still a promiscuous enzyme, and this is the only one which does not have the motif. And so at the moment, my working hypothesis is rather that the motif is involved in or is important for the ancestral activity, so the activation of the AF5A. And I started to do some site-directed mutagenesis and to do some assays again, but at the moment I only have the mutants, but I hadn't have time to do the assays. Okay, but still, so the motif I think is really interesting, and of course I'm also interested in where is the motif located, and so in the three-dimensional structure of the protein. And I started a collaboration with the structural biology group here at University Kiel, and we calculated some 3D homology models. And so I will walk you through this slide. First of all, it's very important, DHS as well as HSS assemble to a tetramer, and this is the biological active unit of both enzymes. And it's a dimer of so it's rather two dimers, and the active site indicated here with the black diamond is located in the interface. Oh, I'm rather okay. Okay, and now here in the middle, this is the surface representation of a homology model from ipomöa alba. And the two subunits, the AB subunits, are color-coded, blue and blue. And also the entrance to the active site is indicated with the arrow. And now I come to the motif. It's indicated here in dark blue. And alba doesn't have the motif. but this would be the position. And this is very interesting because the location of these sites, it is not really, so there's no real connection to the active site. The active site is in the interface here. And they are also not really connected to the substrate channel or the entrance to the active site. And I can only speculate at the moment, but I think it might be another example for epistatic interactions in such in proteins. And it has been shown previously already that mutations outside the active site can still have an effect. Okay. I see that it's rather late. Maybe shall I stop because we wanted to have a 25 minutes talk, right? And it's already half
I think
past.
it's okay. How many more slides do you have? Six more slides.
6 more slides, yeah. Shall
I think we
continue?
started a few minutes late, so it's okay.
Okay. Okay, I will speed up a little bit. So I will skip the sum up, but what is important? I think it's very interesting to see that we have a lot of HSS gene copies in morning glory species. We don't know the function, and we don't know why these genes were retained that long. I mean, 50 to 70 million years is a rather long period. And well, there are many answers to this why question. Maybe they have another function, or they are pseudogenes, or maybe they just survived by chance. And then also multiple characteristics affect the retention time, like the expression strength of a gene, and also protein-protein interactions can also affect the retention time. And in my last slides, I want to focus on very specific protein-protein interactions. And a prerequisite for these interactions that I want to highlight is the organization of the enzymes in homomers. And if such an enzyme is duplicated with all the regulatory elements, then the copies are co-expressed initially. They form their homomers, but of course, as the two genes are identical, they can also form heteromers and physically interact in this heteromer. And via these interactions in the heteromer, the two copies are, also the two gene copies are connected. So if we assume that one of the copies gains some deleterious mutations, then this copy can poison the complex. And thus, one can conclude that initially, after the duplication event, through this pyrrologous interference, both copies inherit the selective constraints of their ancestor. And in case of the duplication of the homospermidine synthase, it would be purifying selection, because deoxyhyposine synthase, the ancestral enzyme, is highly, it's very important. So it's under purifying selection. And indeed, this is something that we found in our selective pressure analysis in the early stage of the duplication. So in the early branches in the phylogenetic tree, we could detect purifying selection. So I think pyrrolog interference might be an interesting phenomenon which can contribute to stabilize duplicates immediately after the duplication. It can protect them from deleterious mutations and increase their retention time of the duplicates. And I think that's it. With this pyrrolog interference, I'm at the end of my talk, and well, I hope you enjoyed it, and I'm happy to answer some questions if there are, or to discuss the results.
Yeah, thank you, Elizabeth. That was a really nice presentation. Now we would like to invite you to ask questions, and we would like to ask students and postdocs to go first with questions, if there are any. And also feel free to post questions in the chat. Okay. In this case, I've got a question. I was wondering, you have identified these motifs in HSS and DHS proteins, and I was wondering if you see the same motifs in the other lineages in which pyrrolidine alkaloids have evolved, or are these completely different?
No, this is the thing, that this motif occurred in all the. Maybe I can go back to this slide here. So the motif is present in the Senecio of the Asteraceae, so in the Senecio lineage, also in the Boraginaceae we find the motif, and in the Apocynaceae. But I think up to now with the morning glories we have the data sets. Usually people screened for the HSS sequence in PA producing species, and then they found the HSS, and these HSS sequences all have the motif. And now with the morning glories we have maybe the most species included up to now in our data set. And there it becomes clear that the motif is not that, so it also is there, but the functional connection isn't that clear. So yeah. Mm-hmm.
Okay, thank you.
So I had a question about the motif too. This motif, you identified that using positive selection as a signature. Is that true?
In the morning glories, yes, that was the start. And then we just checked in the alignment of the other sequences if it's present. And it is present. That's a bit weird. And Tanya Liefschutz, I don't know, we are collaborating on a project so we're studying the evolution in the Apocynaceae, and she was the one who was like, it's everywhere. All the HSS sequences have these change.
Mm-hmm.
Yeah.
Are there other sequences near the active site or motifs or just single residues near the active site that are present in a majority of the HSS or HSS sequences, but not completely ubiquitous, but just present in a majority of sequences and are near the active site?
So I only have the homology models from the morning glory sequences, and they are, I will go back to them. The interface is super conserved, and the active site as well. So in this illustration here now, the differences between DHS and HSS from Ipomöa alba are highlighted in yellow, and you can see even the DHS and the HSS, they don't differ in the active site. And if I would do comparisons also between the different HSS sequences, they hardly differ. So the differences really are mostly restricted to the surface of the protein, which is also something that was described. Of course, the surface can change much easier and faster than the core of a protein. And here only the two orange spots here. Here we have differences between DHS and HSS. So very, very conserved. I think this is something completely different to the enzymes that taught Bachmann last time. I think your sequence identity was much lower between the different enzymes. Yeah.
Interesting.
Super similar, still different function. Very interesting.
Yeah, we have 2 questions there. Yeah, please go ahead, Benjamin.
Hi there, Elisabeth. Yeah, it was a really interesting talk. Really great work. And I guess the discussion on this paralog interference and how it influences gene duplications is really interesting. And I think there's been evidence with the work on hemoglobin that shows that with these duplications, you can actually get new functions with heterodimers or heteromers. So I'm wondering whether you'd consider kind of co-expressing your 2 clades together within the same E. coli and seeing if you're getting any changes in activity with heteromers.
Yeah, I actually really started to do that. I co-expressed HSS and DHS from Ipomyr alba, and it's a bit tricky to check if you really have a heteromeric complex. And so I had the approach that I have the HSS labeled with a histidine tag and the DHS with a strep tag.
Right.
Then I purified the proteins via the his tag, and then I Western blotted my proteins and checked if I find a strep tag.
Right.
So it's a bit complicated, but because they don't, I really don't have another idea how to check if I have a heteromer. I mean, I could do mass spectros, multi-TOF or something like that, but we don't have access to this technology. So I did this very simple experiment, and interestingly, from Ipomyr alba, they too, although I mean, we still can see the homology model, but DHS and HSS cannot form a heterodimer in Ipomyr alba. I also checked Ipomyr. This is a good PA-producing species. There they can. So at the moment, I don't really have an explanation, but I can see that at least there is they differ. If so, some of them might be able to still form a heteromer, but some of them not. And I think it's difficult to change the interface without disrupting the activity. I mean, this is a very special case as the active site is in the interface. Yeah.
Very interesting.
Yeah. Cool. Thank you for the question.
It's okay. Thanks.
If I can just ask a quick question now, your phylogenetic tree reminded me of Benjamin's tree in his nepentolactone paper, whereby the duplicates had longer branches than the sister clade uniformly, and but yet you showed the amino acid sequence identity. I think that's what you showed in your matrix, and what I guess I'm thinking about this is that most of the long branch length must be due to synonymous substitutions. And so if that's true, have you looked at DNDS of the HSS clade as compared to the whatever you're calling it, the deoxyhypocene synthase?
DHS clade, yeah. It's a bit we checked it, yeah. So I didn't. So we what was interesting is that the tree itself, the branches are long and the HSS clade, but still, so maybe the DNDS ratio wasn't different. Only in the very ending branches there we could find positive selection, and then in most branches we didn't have good statistical support. So so it's not a super good signal, only in the Ipomyr. And in these branches at the beginning, so the branches after the duplication event, we had purifying selection. Yeah.
And that's consistent with your model of paralog interference. That one's that's interesting. Well, you know, so the only thing I'm just going to throw out there is in 2012 we did some positive selection studies on our Sabbath enzymes and just got super statistical significance for this one site in the active site. And so we were very excited, mutated the site, changed substrate preference. And now that we've sampled enough enzymes from enough lineages, very dense sampling, we found out that we were completely wrong about that site. It's embarrassing, but I mean, in 2012 we didn't have that many sequences, but the site was under, highly significant under positive selection, functional implications of this change, histidine to methionine, and like I say, now that we've sampled enough sequences, we see histidine did not change to methionine, and so now we're in the process of tracking down what were the changes that led to this substrate preference change. And so I don't know if I'm trying to say be careful with your interpretation of these statistics or what, but just trying to pass along this anecdote along the way.
Yeah. But that's very interesting. So I can completely imagine that the if you have more data, if your sampling is more dense, then you might have different results. And thank you for that anecdote. I will be careful.
Well, and the other thing was that never really made sense to us. Histidine to methionine is 3 nucleotide substitutions, and so that's got to be a pretty unlikely change. And so, like I say, it seems to be the mutational path not taken in our particular case. So, anyway, I like the talk very much. Thank you.
Thank you. Yeah, thanks for your advice to be careful. I usually really try, that's the strategy for the future, is that I do such site-directed mutagenesis. Because the one thing is, okay, positive selection, it's a calculation, and then but it's still a hypothesis, and I try to test this hypothesis with then going back to the lab, doing mutations. And what I did is I mutated, now I have to think because it's a while ago, I think I introduced the motif into the DHS of Hypomyone. And then at least, so changing these 3 amino acids really made this substrate shift. So I could make the DHS HSS. But still, I do have my doubts, as I don't have a clear explanation why this is the case. Even if it's involved in so my theory now is it's just that the A5A is not accepted, and the A5A is a protein substrate, so it has to attach to the enzyme, and there are a lot of interactions, and then maybe subtle changes can really affect the binding of the protein substrate, and this would make more sense than making it optimizing the HSS activity.
Oh, that's cool. No, that's great. Well, and I'll just mention in closing that Benjamin also found positive selection, I believe, in his long branches, and those do seem to have been important. So, I mean, maybe it was just our study, but anyway, thank you.
Maybe you could, so I am not aware of the study of Benjamin. Maybe somebody can put his paper in the chat. Would this be possible?
Oh, yeah.
To share that paper?
I can email it to you after this.
Or that, yeah.
And to be honest, following up on what Todd says, we detected positive selection. We didn't do the mutations, and yet, should we say? And I think that adding the mutations onto this experiment is really important. Ideally, we should have done them maybe, but to kind of verify that the positive selection is having a change in activity. Yeah.
Todd, just to confirm, you said that when you detected that positive selection, you made the mutations and they had an effect, just to confirm.
They were knockouts. Okay. I mean, they completely switched our enzyme from preferring, in this case, benzoic acid to showing near complete preference for salicylic acid, just like we hypothesized. The statistics said this one site, and only this site, in fact, changed on this. What was important, it was under positive selection on the branch we inferred change along. and we made the mutation happen. And now we find out that is just not how it happened. You know, no, these are enzymes that diverged 120-ish, million years ago. So, but at any rate, yeah, it's just been an eye opener for me, and we're working on trying to figure out what really happened. So,
I think there's some debate in the kind of phylogenetics method development literature about these sorts of positive selection analysis and PAML, that sort of approach. So I guess there's a group of people who think they are accurate and work, and others who are not so sure. So there are some, some things like codon usage tables. If you have nucleotides that substitute two at the same time, those things can kind of damage or kind of show positive selection when it might not really be happening. So I think then it can be one part of a story, so to say, rather than kind of a result in and of itself.
Mm-hmm.
If a person were curious about, what you just described, Benji, do you have a suggestion for some reading?
Not off the top of my head. I mean, okay. I'm aware that the original work was done by Zihen Yang, and there's quite a lot of there's some literature about that, and there's some kind of disagreements as well. I can share them with this group, but not right now, because I need to find them.
Yeah, that's fine.
Yeah.
Masatoshi Nei and Shozo Yokoyama really dug in hard against the positive selection, those types of positive selection analyses, and more recently the branch sites models. But quite frankly, when we had our 2012 paper reviewed, I just got so much grief from the reviewers for using those statistics. And maybe it was because we were looking at such an ancient divergence that I don't even try anymore. So anyway.
But so this divergence time is really it doubles the time that I analyze here. When you say 120 million, oh, okay.
Yeah. So that may be part of the problem in our case. Mm-hmm.
Mm. And then also, but I really think it's fascinating that your sequences, they share a sequence identity of, how much did you say last time? 50%? 60%?
I mean, it depends which ones we're talking about, but 60 to 70 maybe, yeah.
Okay. Yeah, cool. I wish we would have more enzymes of this pathway. We would know more of them, because then we come to these interesting questions that was also already discussed last talk. This, like, how do pathways evolve? The patchwork model, I think, is a very interesting model, and what I also hope to do in the future. So this Ipamea alba with the primitive pathway might be an interesting model to study, like, how does the pathway evolve? Because it's a very primitive.
One thing that it's possible, can you go back to the structures of the
enzymes?
Not the enzymes, the substrate, the products. Any slide. So it's very much possible that these are catalyzed by BHDs. The next enzyme might be a BHD acetyltransferase. Because you have some structures, I think Ipamea alba or somewhere, where you have just an OH, and other structures have these acyl chains attached. Yeah, exactly.
Yeah. This
so,
one, yeah.
So this most likely a BHD, I mean, possibly SCPL, but most likely a BHD. And yeah, we worked quite a lot on BHDs, and it's possible to predict in some clades at least if clades in that BHDs in those particular clades might be involved in these sort of reactions. So.
Okay. Yeah, that's interesting. PhDs. For the esterific, so connecting to this hydroxy group via the ester bond, another acid. Yeah. And okay. And the thing is that so we are a small group, we don't have too much money, we really do the sequence identification in our species by PCR with degenerate primers, and then it's very laborious to get the sequences. And I think if we start to screen for candidate sequences, this would be a lot of work. And we are lucky in this case because this is a single copy gene. If we would have enzymes which are part of gene families, oh my gosh, I think we would. I don't know. We think we need more money so that we at least can start with some transcriptome analysis.
Yeah.
Yeah.
But that's what Lars's work has been, and yeah, essentially looking at BHDs and BHD character. How can we predict? Because BHD is a large enzyme family. How can we predict from BHD sequence or phylogenetic position if they would be involved in some substrates? So these look like some substrates that are used in some clades of BHD. So we can talk later about this.
I can remember Lars presented once his he presented this research. Now I just got it.
A while ago.
A while ago, yeah. Yeah, maybe we can get in touch about such ideas. And Lars is coming to Kiel, I heard, and
for Christmas.
so I have the expert here. Yeah, but thanks for the advice to check for the BHD enzymes. Okay.
All right. It doesn't look that we have more questions. Yeah, thanks everyone for being here today. And if you want to ask a couple more questions in a more smaller setting, please stay on the Zoom. But otherwise, thank you very much. And we see us again in it's not 2 weeks. It's November 12th. Am I correct? Oh, it's 2 weeks actually.
It's 2 weeks. Huh?
Yeah, so it's November 12th. We'll have Craig Schenk giving a talk on acyl sugars.
Okay. Yeah, thank you again. And so I stay online a little bit longer to see if somebody else is interested.
Great, thanks very much. Bye.
Bye-bye.
Bye.
Thanks, Ben.